Hey guys, this is our lecture 3 review for lecture. So in this video, I'm going to be going um, over the study guide, as well as where can you find some of the answers, and as well as some answers but not all. Also, just a little disclaimer in this video, this is not an exclusive list of what will be on the test. It's more or less just to aid you in guiding you as to what can possibly be on the test, and then also some potential questions and potential formatting of the question that is fair game for the exam. So let's go ahead and get started. I'm just going to work through the exam kind of like I've been doing in the past. So with the first question, it says, what is a redox reaction? What does it mean to be reduced and oxidized? So that can be found in our chapter 7. So that will be slide 4. So a redox reaction is a chemical reaction of where electrons are being transferred. So whenever something is being oxidized, the substance is losing electrons, and whenever reduction is occurring, then a substance is gaining electrons. So remember that terrible mnemonic, oil rig, oxidation is loss, whereas rig is gain. So that is a redox reaction. Okay, so with that, our next question entails, oops, wrong one. So why does a cell carry out cellular respiration? So cellular respiration, it's the process as to, let me go ahead and just pull it up so we can walk through it together. So, not this one. Is this the right one? No, I'm sorry guys. So cellular respiration. So cellular respiration is the process where cells, specifically in plants as well as animals, where they will break down sugars and turn it into energy for themselves. And then that is when work is performed at a cellular level. So the process of cellular respiration is pretty simple, as we can see below. So this is where glucose and oxygen are the inputs, so that is your reactants, and then your output is carbon dioxide, water, and energy. The most important input and output, so the most important input is glucose, and the most important output is ATP. So, and then the last question, where what would happen to a cell and an organism if a step of cellular respiration were blocked? So big picture wise, what would happen is that the process would not be carried out. It would not be able to go into the next step. But y'all know that each little step of cellular respiration, there are 3. So you would have to tell me specifically, like if I said if something was wrong with the glycolysis process, what would happen, or specifically what would happen in the ETC cycle if ATP synthase wasn't working. So you would need to be a little bit more specific than that question. But big picture wise, the process would not carry on into the next step. Okay, so with that, it says write out the formula of cellular respiration and then make a concept map. So the concept map is a flowchart that we did in class. So below on the screen that you see in the white text box, that is the actual process of cellular respiration. So let me find that slide. So this is the overall process of cellular respiration, where you have glucose that comes in, and then at the very end you have ATP that comes out. So that's the big process. The flowchart that I'm referring to is this document. I handed it out in class. It's also posted under our modules for exam 3. So we did go over this in class, so definitely be able to know the processes forward and backwards, all 3 processes, okay? So you need to know glycolysis, Krebs cycle, as well as oxidative phosphorylation, also known as the ETC cycle. So definitely know that. Okay? And then I believe another question was, what would happen if a step of cellular respiration didn't work? So kind of like the before, but with that, I'm going to go ahead and actually pull up a figure. So with that, I'm going to pull up this figure. So if it'll zoom in even more, I don't think it will. Okay, so with this one, so we have glycolysis. So glycolysis, as we know, is anaerobic, meaning that oxygen is not necessary. So let's say that there is something wrong between the process of going between glucose to pyruvate. So if there was some kind of process wrong right there, then what would happen is that everything downstream of it would not occur. Now, whereas let's say that we had oxygen present, but there was something wrong between the Krebs cycle to oxidative phosphorylation. So let's say that somehow the electrons being like the electron carriers from the Krebs cycle to oxidative phosphorylation wasn't occurring, so then we would say that the ETC cycle would not happen, as well as ATP would not be made. So that's the general idea of what I want you to be able to answer based on that final question on number 3. What would happen if a step of cellular respiration did not work? So I would want you to be able to dissect every little part of it and be able to tell me if I said a specific point in cellular respiration as to what would happen downstream of it. Okay, so with that, we're going to go into what is an anaerobic reaction. So let's go ahead and pull back up our slides. So let me go ahead and find that slide. Okay, so an anaerobic reaction. So this is primarily in prokaryotes, not so much us. So this is where glycolysis will occur because glycolysis itself does not require oxygen present for it to happen. So majority of bacteria will do this, and the fact that they don't have a mitochondria, they do this on their plasma membrane instead. And then instead of using oxygen as their final receptor, they'll use sulfate, which is the SO4. And then from using sulfate, what they'll do is that instead of making water, they'll make hydrogen sulfide. And that hydrogen sulfide, just a little fun fact, it stinks. It smells like, to me, it smells disgusting. A lot of people say it smells like rotten eggs. To me, it smells worse than that. But that's the premise of anaerobic reaction. So this is where oxygen is not present. And then also the fact that it occurs in a good chunk of prokaryotes, so like bacteria. Okay, so with that, the next question is, what is fermentation? So there's 2 different types of fermentation. We're going to talk about lactic and alcoholic. But before we do that, in general, fermentation, it's like cellular respiration in the fact that it begins the same way. So it begins the same way as cellular respiration because they both have glycolysis. But where it differs is that the pyruvate that is made in glycolysis does not continue being oxidized, and the citric acid cycle and the electron transport chain don't run. So instead, what happens is because the ETC is not functional, the NADH made in glycolysis, it can't drop off its electrons back into NAD+. So the fact that it can't do that, there is a additional reaction that will help the NADH to drop off that electron, and in order for it to go back and become NAD+, in order to resupply glycolysis. So that is primarily in both of them, in both lactic and alcoholic, but they kind of do it a little different. So with lactic, what happens is that glycolysis will happen, just like in cellular respiration. But instead, what will happen is that after a pyruvate is made, a lactic acid will be made instead. And then with lactic acid being made, that NADH becomes NAD+ again. and then NAD plus can go back and rejuvenate the whole cycle and start all over again. Some fungi and bacteria can do this. Also, we do ferment a little bit. We're not very good at it. We prefer not to ferment, but this is where you have the pain and the pain sensation inside your muscles, and that is because you have actually lactic acid building up inside your muscle cells, whereas if you had a lot of oxygen present, you wouldn't have it. So with that, I'm going to pull up another figure that shows the lactic acid. So that is this figure right here. So we have the glycolysis process just like normal, where glucose comes in, we make ATP and we make NADH, and we make pyruvate. But instead of the pyruvate entering Krebs cycle and everything, what will happen is that the pyruvate will become lactate. And then in that process of pyruvate becoming lactate, what will happen is that NADH, that came originally from the glycolysis process, will drop off its electrons just like how it normally does in order to become NAD+, and then it will go back and rejuvenate to go back to glycolysis. So it's a constant cycle. So that is for fermentation in the context of lactic acid. Now, if we have fermentation in the context of alcohol, it's the same process where it starts off with glycolysis, but the difference is that instead of the pyruvate being converted into lactic acid, it gets converted into acetaldehyde. And it does that by losing its carbonyl group. I'm sorry, not carbonyl, carboxyl group. And it makes carbon dioxide. So the acetaldehyde, it's converted into ethanol, which is the alcohol part. And then it does that because NADH was made during glycolysis. And we do this a lot whenever we're trying to make beer, make vodka, whiskey, whenever we ferment. Excuse me. With making bread and everything. So we do this process for food-wise for us. Now, oh no, what happened to the picture? Okay, here it is. So, with fermentation in the context, oh, that's not the right image. Ugh, I'm so sorry, guys. We'll just go to the one in the PowerPoint. But what it is that glucose comes in, ATP comes off, NADH comes off as well. Same thing with pyruvate. So that's the same process of glycolysis constantly. Now what's going to happen is that this part in the red circle is going to pop off, and that's going to become our CO2 byproduct. And with that popping off, we get acetaldehyde. And then in the process, the NADH that came from glycolysis will come and be reduced. Will reduce with the acetaldehyde, and in that process, you'll get NAD+ again, which can then rejuvenate glycolysis, and then we will get ethanol. Okay? Okie dokie. So that is alcoholic fermentation. Okay, so now with that, we're going to talk about photosynthesis. So photosynthesis, it says draw a concept map. So whenever I say concept map, I mean flowchart. So same thing. So that is this case study that I handed out in class last week. So this is the concept map and everything. So we did go over this in class, so I'm not going to spend too much time on it. We did go over some case studies and application questions during class time. So with that, I'm going to just leave it right there. And then after that, it says talk about the Calvin cycle. So the Calvin cycle was not shown on this. This is solely the light dependent reaction. So this is only including the photosystems. But with that, let's go ahead and pull up the slide that goes over the Calvin cycle. Which is right here. So the Calvin cycle is in 3 steps. So the first step of Calvin cycle is carbon fixation, and then you have reduction, and then you have regeneration. So definitely I want you to be able to differentiate between those 3 steps, as well as what happens in each step. So with that, I'm going to go ahead and just pull up the next slide and then walk through this figure. So the first step, like I said, is carbon fixation. So that is when carbon dioxide will bind to RUBP. And then in that step, what will happen is that he will become split into the 3-PGA molecule. And it does that with the help of an enzyme called ribisco. And then after we have the 3-PGA molecule, reduction can occur. So what will happen is in the reduction step is that ATP and NADPH are used in order to convert the 3-PGA molecule into a 3-carbon molecule, which is known as G3P. So what will happen is that since we had 6 PGA molecules that started off, and then after that we're going to end up with 6 GP3s, but what's going to happen is that only one of those GP3s is going to be destined to go and make glucose, whereas the 5 other ones are going to enter the regeneration step. So this is where some of those GP3 molecules, they are recycled, and then they're recycled in order to rejuvenate that RUBP molecule. And then once that RUB molecule is regenerated, then it can bind again with carbon dioxide with the help of ribisco. And then it just constantly goes in a cycle. So that's why it's called the Calvin cycle. Okie dokie. So with that, I do want you to, I'm going to show you some intermediate steps of the Calvin cycle. So this is the Calvin cycle as a whole, which is what we just saw on our slide. So right here, this is the actual step of how we get that 3-PGA molecule. So we have the RUBP, it will combine with carbon dioxide, so that is why we have a 6-carbon. And then what will happen is that it'll get split down the middle. And if it gets split down the middle, then we're going to have 3 carbon molecules with the phosphorus, and then 3 carbon molecules with the phosphorus. So that's the carbon fixation process, whereas this is the reduction process right here. So what happens is that the 3-PGA molecule, ATP will come in, and then that will become ADP. So it phosphorylated it, so it added another phosphate group to it. And then in that process, what it does is that NADPH will come in and dephosphorylate it into GP3. So those are the intermediate steps. I just wanted to show you just so you had a little bit better idea of what's going on. So definitely know the Calvin cycle. Okay. So with that, that brings us to what kind of organisms can do photosynthesis. So pretty much any organism that is photosynthetic can do it. So meaning like plants, trees, some cyanobacteria. So cyanobacteria are bacteria that are photosynthetic. Protists, some of them. But just know that mammals themselves are not photosynthetic, essentially. Okay. So with that, the next question was, how can an herbicide kill an organism? So it kind of goes back to our case study that we went through in class. So an herbicide, blanket definition is it kills plants, essentially. But what herbicides do specifically is that they inhibit processes of photosynthesis. So kind of like what we did in class where different herbicides, they target different parts of photosynthesis. So if you target one part of photosynthesis, what's going to happen downstream of that? I do want you to be able to, what's it called? Apply that to some questions on the test. So they're going to be kind of similar to what we did in class. So if you have any questions, definitely ask. But the big picture of herbicides, what it does is that it is photosynthesis inhibiting. So that means that it'll block somehow, some way photosynthesis. The main way that it'll do it is by blocking the transfer of electrons. And without the transfer of electrons, the energy that was absorbed from the sun cannot be transformed into energy that is usable for the plant. So meaning that they can't transform it into ATP. And if they can't transform that energy, they can't undergo photosynthesis, what will happen is that the plant is not able to like regenerate new tissues or anything if necessary, like meaning that they can't continue to grow leaves, as well as if they can't continue to grow leaves or even do photosynthesis, then unfortunately, if that herbicide is not removed from its environment, it's not going to be able to sustain life. Okay, so with that, that's going to bring us into the differences between CAM 3, I'm sorry, not CAM 3, C3, C4, and CAM photosynthesis. So let me go ahead and pull up that slide. I'm just going to bring up this one. So actually, I'm going to just pull up these pictures. So that brings us to these guys. So these 4 figures, I'm sorry, 3 figures. I can't count today. So in these figures, I'm going to walk through them. So in a CAM 3 plant, so that's the first one, the first little green panel. So this is like a normal plant. This is one that doesn't need any adaptation to do photosynthesis because of its environment. So it'll go through the first step of the Calvin cycle, which is the carbon fixation by Rubisco. It'll go through the process just like normal, what we've already talked about. So those are the processes that we've been talking about the entire time. Whereas C4 pathway, they need an adaptation. So what happens is that the light dependent reactions that we talked about, excuse me guys, sorry, that we talked about, like with the photosystems and everything, that has to occur separately from the Calvin cycle, as you can sort of see here. So the Calvin cycle, what will happen is that it will occur inside a bundle sheath cell, whereas carbon fixation, it will occur somewhere else. So generally what will happen is that carbon dioxide will enter. So in C4 plants, their stomatas are only partially open, they're not completely open, so they only allow a limited amount of carbon dioxide to enter. But whenever carbon dioxide does come in, what's going to happen is that carbon dioxide is going to be fixed at first in the mesophyll cell, and it's going to be fixed in the with the help of a peptase enzyme. And then what will happen is that after it's been fixed, it will be fixed to a 4-carbon molecule. Oh, sorry guys, I'm so tired. So after carbon dioxide has been fixed, what will happen is that it will be exported into a bundle sheath cell, and then from there it will undergo the Calvin cycle like normal. Okay, so that is for the C4 plants, whereas for the CAM plants, they're definitely a lot different compared to the other 2 that we've discussed. But with CAM plants, what happens is that they are in extremely dry areas, such as like a desert. So their stomatas, they're closed completely during the day in order to ensure that they don't lose a lot of water. So with that, what happens is that they don't undergo, they don't uptake their carbon dioxide until nighttime. So once it's nighttime, their stomatas will open, carbon dioxide can enter, and then just like in the C4 pathway, it'll also be fixed to a organic molecule using the peptase enzyme. And then after that point, what will happen is that as daytime comes around, so once daytime comes around, that carbon dioxide is trapped inside of the plant cell, and then that carbon dioxide will then be able to enter the Calvin cycle, because Calvin cycle does not occur until it is daytime. So while they are constantly fixing these carbon dioxides, there is an abundance of carbon dioxide that is occurring inside the cell. And then once daytime occurs, then what will happen is that they can undergo the Calvin cycle. Okay. Yep, I believe that's the big picture that I wanted to touch base on. And then with that, it says to like make a table and everything. So here's a table. This is one that is from our lecture. I added a little something else to it just to make it a little bit easier. But these are examples of the plans for each type of pathway that we talked about, as well as how do they fix carbon, what enzyme do they use to fix carbon, and then also why do they have what they have, and it all comes down to water loss. And then also what cells are involved. So C3s and CAMs, they primarily use the mesophils, whereas the C4 uses the bundle sheet cells. Okay. Okie dokie. So I believe with that, that will bring us to the different roles of mitosis. So the different roles of mitosis. Let's open up chapter 9. So the different roles of mitosis. So the ultimate goal, I don't know why this froze. Sorry guys, hold on. Okay, there we go. Okay, so the key roles of cellular division, which is also known as mitosis. So for unicellular organisms, so we are not unicellular, so this has to do with like bacteria, some yeast are considered unicellular, but the whole purpose of them is in order to produce more of themselves, essentially. So bacteria, they don't undergo sex, they don't have sexual reproduction, so their form of mitosis is called something binary fission. And then with yeast, what they do is that they bud, so it's kind of like binary fission, except for the fact that instead of it being completely the same size and everything, it'll be a little bit smaller, the bud, which is the reproductive cell that was just produced, the progeny. But for unicellular organisms, the whole purpose of being able to undergo cellular division is to reproduce an entirely new organism. Whereas multicellular eukaryotes, such as ourselves, we do this so we can actually renew and like repair or completely replace cells as needed. So it's a little bit different. Okay, so the next question then asks, what is a genome and how does it relate to chromosomes such as DNA? So let's find I believe this is, nope, next slide. There we go. So a genome is a complete set of genetic information. And then what it does is that it provides essentially a blueprint of information as to what is the whole function of a certain cell. So like what is the function of a stomach cell? So we have a gene that will code for saying that a stomach cell, his whole purpose is to produce acid in the stomach. Or like a muscle cell, the whole purpose of a muscle cell is to be able to contract and relax, and so on and so forth. So in living organisms, the genome, it's stored in a molecule that is called DNA. And then a chromosome is one DNA strand wrapped tightly around histones, which is this. So we have a whole chromosome, so that's the big picture. Within that chromosome, we have these chromatin fibers. And these chromatin fibers, what they do is they wrap around these histones, which are, you can think of them literally as stones, where they aid in being able to have our DNA so condensed in one area, because our DNA is extremely long. And you'll actually kind of see that in class a little bit. It's kind of cool. And then from there, you'll get your genome, which is your DNA, if you unwound it. We'll go more into detail in class about that. But just know that, know how to define a genome. Know that the genome is stored in a large molecule that is called DNA, and then more accurately, it's stored in chromosomes, in the format of a chromosome. Okay, so with that it says, make a chart of the phases of interphase, mitosis, and cytokinesis. So let's actually just talk about the three parts, those three parts of the cell cycle individually real quick. So let me find that slide right here. So it talked about interphase. So interphase, that is the whole phase of the cell cycle where the cell grows and copies itself in preparation of cellular division. And then mitosis is the actual physical division of the chromosome or of the genomic information. And the cytokinesis is the division of the cytoplasm. So this is where you'll eventually get a whole new cell at the very, very end. Okay, so with that, I'm gonna pull up the table. So right here is the table of interphase. So interphase has three main phases. It's G1, S phase, and G2. So in G1, what's going to happen, so this is in order, guys, so this is a timeline. So G1, what happens is that the cell will grow in size, so it'll get bigger. And then whenever it gets bigger, what it does is that it is increasing the amount of cytoplasm. And then also it is making copies of all the organelles inside of the cell. And then from that, what happens after that is complete, S phase will occur. And this is where the DNA is actually replicated and is copied. And then after the DNA is completely copied, we'll enter G2. And this is where the cell is actually preparing itself in preparation of mitosis, which is cellular division. And then also is checking the chromosomes for any errors that may have occurred. And we'll talk about specifically the checkpoints as to how do we check to make sure that our DNA is free of error. We'll talk more about that later. Okie dokie. And then it says to do it for mitosis. So mitosis is the following right here. I'm actually just going to walk you through a figure. So I actually really like this figure below. And it's really, really blurry. Great. 1 second, guys. Let me see if I can make it not blurry. Okay, so I wasn't really able to make it less blurry other than in this picture. But let's go ahead and just walk through mitosis. So mitosis has 5 main stages or phases. And you can remember it as PP-MAT. So it goes prophase, prometaphase, metaphase, anaphase, and then telophase. And then following telophase, you have cytokinesis, which is not part of mitosis. It's kind of its own little step, and we'll talk about that later. But let's go ahead and walk through this entire process. So what happens is that at the end of G2 phase, which is where we ended, what will happen is that you'll enter something called either early prophase or just prophase in general. So in prophase, what happens is that the chromatins or the chromosomes, what they do is that they start to condense. And then what will happen is that microtubules or microspindles, whichever one, what they will start forming inside the cell. And then the mitotic spindles, what they will do is that they will eventually pull the chromosomes apart. And then also the nucleolus is completely gone at this point. Okay, and then from there you'll enter prometaphase, which is also known as late prophase. Either one, if you say late prophase or prometaphase, I'm okay with either one. I learned it as late prophase. I'm really sorry, guys, it's a little blurry. But what happens is that the nuclear envelope completely gone. And then what will happen is that the microtubules, they'll start to extend, and they'll extend to the chromosomes and attach to the chromosomes. And then the microtubules of the spindles, but they will attach specifically to the point of the kinetochores, I believe that's how you pronounce it, of the chromosome, which is essentially at the center of the chromosome. So it's happening in prometaphase. And then, so this is kind of just a zoomed in version of it. So we have a chromosome, and then the kinetochore, it's at the very edge of the chromosome in the center. Okay? Okie dokie. So with that, we're in metaphase. So this is where the chromosomes are completely lined up in the center. The assembly of the spindles they're completed, meaning that they're all attached and everything. They're completely formed. Now all the chromosomes are attached to their respective spindles at their kinetochore. And then, like I said, they line up in the center, and whenever they're lined up in the center, it's called the metaphase plate. So that's metaphase. Now we're going to go to anaphase. So that's the next one. So anaphase is where you have cohesion proteins that hold together the sister chromatins. So originally, so if you see back here, it kind of looks like an X. So there was cohesion proteins that were present that were holding it together. But now what happens is that those proteins, they're cut. So as you can see, like now there's a split in between the sister chromatins. And then once that split occurs, the microtubules will start pulling the sister chromatins apart. And chromosomes are now being pulled to their respective poles of the cell. So they're being pulled towards these, they're called asters. They're being pulled towards those little orange little chiclet things, and those are the asters. Now after anaphase, what will happen is that you'll enter telophase. So telophase is where two daughter nuclei form, meaning that you're starting to reform the nuclear membrane. The spindles, they will detach, they'll break down. And then the chromosomes, they start to decondense. And then at that point, you enter something called cytokinesis. So cytokinesis will start beginning. And depending on what cell type you're in, whether it be animal cell or plant cell, will determine the type of cytokinesis that will happen. So if you're in an animal cell, what will happen is you'll start pinching off your cytoplasm, so it'll start kind of squeezing in, and then that is called cleavage, or the cleavage furrow. So there's starting to become like a groove between the cells. So at first you have like, if you put your hands together to make like one big ball, but then you curl in your fingers to like start making a fist on your respective hands, that is you forming a cleavage furrow. And then eventually what will happen is that the plasma membranes will pinch off, and then you have two separate cells. Now, whereas in a plant cell, what will happen is that a cell plate will form during cytokinesis, and vacuoles, what will happen is that they'll deliver materials to the respective area and deposit the necessary needs in order to build a new cell wall. And then after the cell wall is completely built, and it separates two whole new cells. So overall, at the end of mitosis, what happens is that you have two new diploid daughter cells that are identical to each other. So it's like having identical twins. They're the exact same at the end of mitosis. Okay, so with that, we only have a few more questions. I believe the next question, let me see. What conditions must be met for the 3 different checkpoints of cell cycle? So where it says add to diagram below, it's talking about specifically this diagram. We'll go over that whenever we get to this diagram. But for now, let's talk about those checkpoints. So these checkpoints, there are 3 of them. So there are 3 checkpoints, like I said. Actually, let me pull up the figure. It's a little bit better. Okay, so there are 3 checkpoints. There is the G1 checkpoint, G2, and the M checkpoint. So the first one, the G1 checkpoint, oopsies. So the G1 checkpoint, what it is, I'm going to just pull up that figure. So what it is that it occurs during the G1 phase of interphase. For most cells, the G1, it seems to be the most important for most cell types. But what it does is that it'll determine if the conditions to divide are correct, meaning that is it right to go ahead and continue the cell cycle? So what will happen is that if there's enough resources and the cell is big enough to divide, it'll get the go ahead signal, and then it'll go ahead and enter into S phase so it can replicate the DNA. Or if somehow the conditions they're not correct, then what will happen is that there will be a stop signal, and the signal will send the cell essentially backwards. Okay? And then the next one is G2. Oh, I don't have a picture for G2. Sorry, guys. So this happens at the very end of the G2 phase of interphase. So this is the one that controls the entry into the mitotic stages. And what it will do is that it will check the chromosomes to make sure that they were replicated correctly. So if the chromosome, they are damaged, then they will attempt to repair. If the cells cannot be repaired, then the cell will undergo programmed cell death, which is apoptosis. But if the genetic material is deemed fit for duty, then the cell will continue on into mitosis. So that's the G2 cycle. Now with the end checkpoint, this happens during metaphase. So this is where we're already in mitosis, but this happens during metaphase, and it's at the very end of metaphase. But what it does is that it makes sure that the sister chromatids are actually attached to the spindles. So if there's no attachment of spindle fibers to the chromosome, then it will tell the cell that it's stuck and it won't continue on and it won't enter anaphase. Also, what it also checks for is to make sure that the chromosomes, that they are actually lined up in the center of the cell to ensure that equal parts of the DNA enters to each pole. So if you have, let me get my pen. So let me make this bigger. Actually, let me just go onto this page. So what it is essentially is that, let's say that you have a cell, and then you have your chromosomes. So I'm just going to do 2 because it makes it easy. You have your asterids over here, and then you have your, that's not attached to the center. Then you have your spindles attached. So it wants to make sure that it's lined up in the middle so that once it starts pulling apart, what will happen is that you will get equal parts of the genome being pulled to the poles. Okay? Whereas if they weren't lined up in the middle, let's say that it was more, that was a little bit more asymmetrical. Oh, that's a terrible cell, sorry guys. Let's say that one was closer to the other pole. So what might happen is that, yes, it may be attached, but since it's not in the middle, what may happen is something like this. So this whole chromosome maybe end up pulling to this side of the pole. So instead of having an equal part of 50-50 DNA like over here where it is 50-50, you only get 25 to 75, which is not good. You don't want more genomic information in a cell than necessary. You want to have the perfect amount. So that is what the M checkpoint does. So if you get this 50-50, then what will happen is that it can enter into anaphase. If you don't, where you get more of the 25-75 ratio, what will happen is that the cell, it will become stuck and it won't go any further. Okay. So with that, let's go ahead and talk about the last, I believe, almost last question. So it says, give an example of the internal and external controls of the cell cycle. So internal controls are actually something that come from within the cell. So an example of that would be the M checkpoint. So specifically with that one, anaphase, it won't begin if anything is wrong. So if there's anything wrong, it won't enter anaphase. In order to make sure that attachment of all of the kinetochores, what they'll do is that they'll activate a complex of proteins, and a lot of times it's known as septarase. I think that's how you say it. And then what happens is that specifically that septarase will break down the cohesion factors that were between the sister chromatins. And then once that cohesion is broken apart between the sister chromatins, then anaphase can occur. So the two regulatory proteins for this is CDK. So he's always constantly around. I'm not really 100% too concerned about you knowing that. It's kind of just more like a little side note, I guess. Okay, so with that, let's talk about external controls. So external controls, these are the ones that come from signals from outside the cell. And one of the big ones is our population density of a cell. So if the population is, if there's too much cell growth, so like if you start coming in contact with neighboring cells too much, it can stimulate whether or not if you should stop or start the cell cycle. And then proteins are released by the cell that will actually stimulate other cells. So if you want to make more cells, then what you'll do is that you'll actually start secreting out these growth factors so you can actually start growing more. A big example of this is the platelet-derived growth factors, so PDGF. This specifically is found in fibroblasts, and these are found in connective tissues such as like collagen. Fibroblasts are extremely important in healing. You'll probably get more detail if you take anatomy and physiology about fibroblasts. But specifically, you will get an influx of PDGFs if you have an injury. And specifically, this growth factor, what it does is that it will stimulate a certain division of fibroblast cells in a culture and in your body. So if you have an injury, you want to repair that injury. So then that is where this PDGF comes in. There's also, I believe I have another slide. Yep. So there's also what animal cells do. We do this a lot, is the anchorage dependence. So this is where a cell has to be attached to a surface in order for it to divide. So if there's no attachment, there's no division. So if you can't anchor yourself, you can't divide. And then, like I mentioned before, there's also density dependence and inhibition. So this is dependent on, you get so many cells to divide, but then once you start realizing like, oh crap, I'm running out of room, the cell will start coming in contact with each other. It's also called quorum sensing, where cells, once they touch each other, they'll be like, oh, I don't need to divide anymore. So it has more to do with population density for the last bullet point. And bacteria do that a lot, so do fungi. Okie dokie. So with that, I'm gonna go ahead and pull up the last image, actually last two images on our exam review. So I've numbered these. You don't see that on your exam review that I posted, but I do want you to be able to tell me the differences between these guys. So number 1 is the sister chromatins. Number 2 is chromosome. Number 3 is the kinetochore. Number 4 is the centrosome. So that is the very, very center of between chromosomes. And then number 5 is the homologous chromosomes, because they're the same, they're pairs. And then number 6 is the microtubules, or the spindle fibers. Okay, so with that, Oopsies. Here's also another nice little figure. It's not the same one that's on your exam review, but it's very, very similar. I actually like this one a little bit more because it kind of shows you what is going on in each stage rather than it being blanketly labeled. But definitely be able to tell me what is A, B, C, and D. Definitely be able to tell me that. And then also on top of that, be able to add in those checkpoints that we talked about. So the G1, the G2, and the M phase checkpoints. So be able to add that onto the cell cycle. Okay, so with that, I believe that wraps up our exam review. If y'all have any questions, definitely don't hesitate to ask. Also, just remember that this review video and the study guide given is not an exclusive list of what is fair game on the exam, but definitely it does cover majority of the concepts that I would like you to know for the exam. So with that, if there aren't any questions, I will see you in class.